Now students, let us take a glance and how many ATP molecules and NADPH molecules are utilized here in the synthesis of hexose, and based on that we follow few numerical based questions. So, we will continue in that way. Okay. So, may you follow the detailed mechanism of Calvin cycle with this. So, Calvin cycle which is occur in all photosynthetic organisms, universal dark phase reaction, and the only way for carbon dioxide reduced to carbohydrate with 3 steps: carboxylation, reduction and regeneration. Regeneration of the acceptor, that is RUBP. Students, now let us follow the energetics of Calvin cycle reaction, and here for the synthesis of 1 hexose let us calculate. For the synthesis of 1 hexose and fixation of 6 CO2 molecules should involve, and the acceptor 6 RUBP molecule should be involved. So, fixation of 6 CO2 molecules by 6 turns of Calvin cycle we consider. Finally, synthesis of 1 hexose we followed. For that, how many ATP are utilized? As already you followed the detailed mechanism, 12 ATP molecules are utilized in reduction phase for phosphorylation reaction, and 6 ATP molecules are utilized in regeneration of RUBP from RUMP molecules. So, totally 18 ATP molecules are utilized in 6 turns of Calvin cycle for the fixation of 6 CO2 into 1 hexose. And here the reducing power involved is NADPH, and how much reducing power utilized, and that is for the reduction of that PCA molecules, 12 NADPH molecules, that is the reducing power utilized in 6 turns of Calvin cycle, that is for the fixation of 6 CO2 for the synthesis of 1 hexose. So, now students, let us count and how many ATP and NADPH molecules are utilized for the fixation of 1 CO2, that is for the synthesis of 1/6 of hexose. Of course, it may not exist, but even then just for calculation we are following, and for the fixation of 1 CO2, that is for the synthesis of 1/6 of hexose, how many turns of Calvin cycle? Only 1 turn of Calvin cycle we consider. And what is the assimilatory power utilized here is, and we can say 3 ATP molecules and 2 NADPH2 molecules are utilized. Okay. So, 3 ATP and 2 NADPH molecules are utilized for the fixation of 1 carbon dioxide, that is in 1 turn of Calvin cycle for the synthesis of 1/6 of hexose, and this is considered as unit of assimilatory power. So, whatever the assimilatory power utilized for the fixation of 1 CO2 molecule mean utilized in 1 turn of Calvin cycle is known as unit of assimilatory power, that is 3 ATP and 2 NADPH. But for the synthesis of 1 hexose, we followed 18 ATP molecules and 12 NADPH are utilized in 6 turns of Calvin cycle, that is for the fixation of 6 CO2. Okay. And during the process we observed 6 RUBP involved as acceptors and 2 DHAP as net gain exported to cytosol for the synthesis of that hexose molecule, and DHAP is a triose. So, 2 such trioges are involved in the synthesis of hexose, and total 12 G3P molecules we observed in the pool, and that is in 6 turns of Calvin cycle, and 6 RUBP molecules the same number regenerated, and for the regeneration of that 6 RUBP, number of xyluloses involved 4, number of riboses involved 2 accordingly, and we get questions in objective orientation. So, with this we can follow the energetics of Calvin cycle. Okay. But now let us do one more. Based on this information, let us try, can we calculate what is the assimilatory power utilized for the synthesis of 1 sucrose molecule. Here just I change, instead of 1 hexose, if I mention sucrose, can we calculate the number of ATP and NADPH and as well as number of CO2 fixed and number of RUBP involved. Okay. Let us try, and here sucrose is a disaccharide, you know, is a 12 carbon with 2 hexoses, that is glucose and fructose. So, just consider sucrose which is a disaccharide with 2 hexoses. So, now let us calculate accordingly. For the synthesis of 2 hexoses, how many ATP molecules, and that is 18 into 2 should be multiplied with 2. So, totally 36 ATP molecules utilized, and how many NADPH? 24 ATP NADPH molecules are utilized. So, and accordingly we can calculate for the synthesis of a sucrose molecule. Why we are discussing sucrose here? Because most of you know that phloem translocate the food material in the plants in the form of sucrose. Of course, may hexosynthesis happens initially, but finally that should be converted into sucrose for translocation purpose. So, we are calculating the assimilatory power required for the synthesis of sucrose. Okay? And sucrose mean 2 hexoses we consider, that is glucose and fructose, and for that 36 ATP molecules utilized and 24 NADPH is the reducing power utilized, and 12 molecules of carbon dioxide should be fixed. Okay? So, how many turns of Calvin cycle now we consider? 12 turns of Calvin cycle for the synthesis of one sucrose molecule. Accordingly, we can calculate whatever the count is given. Okay, students? Okay, students. Now, let us discuss the next subtopic in dark phase reaction, that is C4 pathway, in other words called Hatch-Slack pathway. Already we came to know that C3 cycle is universal, that occur in all photosynthetic organisms. That is the way how CO2 reduced to carbohydrate we followed. But now this C4 pathway, and which is also called Hatch-Slack pathway, is an alternate pathway for the fixation of carbon dioxide that occur only in C4 plants. So, based on the dark phase mechanism, dark phase reactions, we categorize the photosynthetic plants into two types, that is C3 plants and C4 plants. C3 plants mean that perform only Calvin cycle reactions during dark phase. Okay? But C4 plants mean the plants that perform C3 cycle of course, but additionally to supply more carbon dioxide time to time for Calvin cycle reactions, they follow a special and additional pathway for CO2 fixation, and that is called C4 pathway. Such plants that perform C4 pathway for additional fixation of carbon dioxide as well as Calvin cycle for reduction of carbon dioxide into carbohydrate, they are called C4 plants. And as we follow the examples for C4 plants, and they include sugarcane and maize that come under monocots, but not all monocots. Here we have to memorize the examples. It is not a family character, it is not a group character, and specially the scientists, they traced out this C4 pathway in certain plants only, and here the examples mentioned already traced out in sugarcane and maize, as well as some dicots like amaranthus and tribulus recently identified with C4 pathway. So, C4 plants, they perform C4 pathway as well as C3 cycle. The additional pathway for CO2 fixation is C4 pathway, and the examples for C4 plants include both dicots and monocots we mentioned. Initially that is observed only in monocots like sugarcane and maize, but later it is also found in amaranthus and tribulus, such dicots. Okay. So, now and let us follow, there is a special kind of leaf anatomy found in C4 plants, mean the plants that perform C4 pathway, they show a special kind of leaf anatomy that is known as cranch anatomy. Okay. So, now let us focus on cranch anatomy, which is the special kind of leaf anatomy found in all C4 plants and only in C4 plants. Cranch anatomy is observed in all C4 plants, whether they are monocots or dicots, and observed only in C4 plants, that is to perform C4 pathway. Okay. Now, let us see what is cranch anatomy. Here this is the leaf anatomy of C4 plant I displayed here, and we follow that upper epidermis, lower epidermis, and in between the mesophyll area, and this is the vascular bundle, and the vascular bundle surrounded by bundle sheath. So, as we focus on bundle sheath and mesophyll of C4 plants, and that show cranch anatomy in their leaves, both bundle sheath cells and mesophyll cells, they bear chloroplast here. Actually, in normal photosynthetic plants, maybe compare with C3 plants, only mesophyll cells bear chloroplast and perform photosynthetic mechanism. So, dark phase occur only in mesophyll cells in C3 plants, but dark phase occurs in both mesophyll and bundle sheath cells in C4 plants. So, here one special feature is bundle sheath cells also bear chloroplast, and both bundle sheath and mesophyll cells involved in dark phase reaction. As we verify the chloroplast of bundle sheath and mesophyll, the chloroplast of bundle sheath are bigger in size and they are agranule chloroplasts. Bundle sheath chloroplasts are agranule, chloroplast mean without grana, agranule chloroplasts, and they do not show grana, so they do not perform light phase reaction and they perform only dark phase reaction. And mesophyll cells, they are with normal chloroplasts, that mean granule chloroplasts, and that perform both light phase and dark phase. So here the dark phase mechanism in C4 plants occur in both bundle sheath and mesophyll cells, and they bear chloroplasts, but there are two types of chloroplasts we observe. Bundle sheath chloroplasts are bigger and agranule, and mesophyll chloroplasts are normal and small in size, mean the normal chloroplast with both grana and stroma and small in size. So can we say chloroplast dimorphism for this? So the plants that show C4 pathway, they show cranch anatomy, okay, and they are with dimorphic chloroplasts, and both bundle sheath and mesophyll with chloroplasts and involved in dark phase mechanism. But as we verify the word cranch, and here cranch anatomy refer the special kind of anatomy where the bundle sheath is with bigger cells and compactly arranged around the vascular bundle in the form of wreath. And cranch is a German word, and that represent that wreath like arrangement, and here that bundle sheath in such kind of leaves as wreath like, so the name cranch anatomy, wreath like anatomy. So which part represent that wreath like arrangement? Bundle sheath. And here both bundle and mesophyll cells, they contain chloroplast and involved in dark phase mechanism, that is cranch anatomy, specially associated with C4 plants. So now let us focus on the detailed mechanism of C4 pathway. C4 pathway also called as Hatch-Slack pathway, because this particular pathway traced out by the scientists Hatch and Slack. So based on their name, that is called Hatch and Slack pathway. And but why C4 pathway? Why it is named so C4? Because during this pathway and due to the fixation of carbon dioxide, the first product formed is oxaloacetic acid, which is a 4-carbon compound. So as the first product formed is a 4-carbon compound during the pathway, this is named as C4 pathway. Now let us follow that biochemical reactions included in C4 pathway. Already we mentioned that C4 pathway is an alternate pathway for only CO2 fixation but not for reduction. And here in this pathway, and time to time carbon dioxide supply will be enhanced. So C4 plants we may consider as biologically more evolved and better adapted for C4, that means photosynthetic mechanism, and time to time they supply carbon dioxide to Calvin cycle reactions very efficiently. So they efficiently perform photosynthesis and they are considered as more evolved in photosynthetic activity with this alternate pathway of CO2 fixation. But now that CO2 fixation, how it happened, and now let us see. Here already we mentioned dark phase reactions occur in both mesophyll and bundle sheath cells, and there is a biological compartmentation we can follow during dark phase mechanism. So here the total dark phase mechanism of C4 plants we represented that occur in both mesophyll and bundle sheath, but whatever the reactions occur in mesophyll, now let us see. In the mesophyll cells, of course this is chloroplast and this is cytoplasm, and there in the cytoplasm a 3-carbon compound phosphoenol pyruvic acid is there, and that is the acceptor, the primary acceptor of carbon dioxide in the mesophyll cells, accept carbon dioxide in the form of bicarbonate ions catalyzed by the enzyme peptase phosphoenol pyruvic carboxylase. So this is the first fixation or primary fixation of carbon dioxide that occur in the cytoplasm of mesophyll cells. As a result, a 4-carbon compound is formed, that is oxaloacetic acid, and this is primary fixation of carbon dioxide occur in C4 plants in the mesophyll cells. And now this oxaloacetic acid now entered into chloroplast of mesophyll and converted into malic acid, another 4 carbon compound by reduction process, and now this 4 carbon malic acid added to bundle sheath cells. There in bundle sheath chloroplast, malic acid, that 4 carbon acid, undergo decarboxylation and converted into pyruvic acid, which is a 3 carbon organic acid, while evolving carbon dioxide. So, whatever the carbon dioxide fixed in mesophyll cells and that entered into bundle sheath in the form of malic acid, but malic acid there undergo decarboxylation and again release that carbon dioxide. Now, that carbon dioxide is available to Calvin cycle reactions that occur in bundle sheath. Now, the carbon dioxide released from malic acid accepted by RUBP, the acceptor of Calvin cycle reactions, and involved in photosynthetic carbon reduction, and so that DHAP formation as usual that we follow in Calvin cycle, and here the DHAP molecule synthesized in the Calvin cycle in bundle sheath cells exported to cytosol of bundle sheath and involved in the synthesis of hexoses. So, as we verified the process here, and in mesophyll cells there is CO2 fixation, in bundle sheath cells also there is CO2 fixation. In mesophyll cells and only CO2 fixation but not reduction, but here in bundle sheath cells the carbon dioxide fixed by RUBP and involved in photosynthetic carbon reduction and involved in the synthesis of hexoses. Okay. So, there is a biological compartmentation of dark phase reactions we observed, and mesophyll cells perform only fixation, but bundle sheath cells perform Calvin cycle reactions and perform reduction of carbon dioxide. But why plants prefer that primary fixation in mesophyll cells is to supply carbon dioxide efficiently every time for Calvin cycle reactions, so that with C4 pathway, C4 plants are photosynthetically more evolved and more efficient, and further they maintain this alternate pathway for CO2 fixation. Now, let us see how the primary acceptor phosphoenol pyruvate is regenerated, and whatever that pyruvic acid released in bundle sheath cells again transported to mesophyll cells, thereby utilizing some extra number of ATP. For one molecule 2 ATP extra utilized and converted into phosphoenol pyruvate. So, phosphoenol pyruvate is regenerated and added to mesophyll cells again, and the process goes on continuously. So, and as we follow this total biochemical pathway of C4 pathway, and we followed that in C4 plants, dark phase mechanism occur in both mesophyll and bundle sheath, and in mesophyll cells only fixation, but whereas in bundle sheath cells that carbon dioxide and fixed and reduced and involved in the synthesis of hexose. And the venue of Calvin cycle reactions is bundle sheath, and mesophyll is only for fixation, that is alternate pathway for CO2 fixation. And here as we verified 2 compartments, CO2 fixed twice, and the first fixation or primary fixation occur in mesophyll area catalyzed by the enzyme pepcase, and the acceptor is a 3 carbon compound phosphoenol pyruvate, and the product formed is a 4 carbon acid OAA. But whereas the ultimate fixation we observed in bundle sheath, and here that carbon dioxide is fixed in bundle sheath of course, and the acceptor is RUBP and the enzyme involved is RUBP carboxylase. Okay? That is the carboxylation enzyme involved in Calvin cycle, and there the carbon dioxide not only fixed and later undergo reduction, and DHAP molecule formation and hexosynthesis takes place in bundle sheath cells. And that is about that C4 pathway performed by C4 plants, and C4 plants show both C4 pathway and Calvin cycle during dark phase reaction, and C4 pathway is only for CO2 fixation and to maintain time to time CO2 fixation and to maintain higher CO2 levels internally to make that Calvin cycle reactions more efficient. Okay? So, that is about C4 plants that perform C4 pathway as well as Calvin cycle, and venue of Calvin cycle is bundle sheath cells, and DHAP formation bundle sheath cells, hexosynthesis also bundle sheath cells. Okay? And that primary fixation occur in mesophyll cells. Primary acceptor is phosphoenol pyruvate, and catalyzed by peptase, and final acceptor is RUBP and catalyzed by Rubisco, that is RUBP carboxylase. Okay, students, now let us know about a special biochemical pathway that occur during dark phase reactions in succulent xerophytes with scotoactive stomata, and that is called CAM pathway. As we follow the full form of CAM, that is crassulacean acid metabolism pathway, because that is identified as a acid metabolic pathway first in crassulacean members. So, as it is traced out in crassulacean members, so crassulacean, as it is related with the formation of acids, acid metabolism, it is named so crassulacean acid metabolism, because they identified formation or accumulation of acids during night time, that is acidification process during night time in succulent xerophytes that shows scotoactive stomata, and deacidification process observed during daytime. So, this kind of mechanism, that is CAM pathway, that can be referred as crassulacean acid metabolism, and specially seen in succulent xerophytes, and that too they are bear scotoactive stomata. So, remember, and the plants with scotoactive stomata, and they perform this special biochemical pathway or special dark phase mechanism, that is crassulacean acid metabolism, that is CAM pathway. Now, let us follow what are the examples for succulent xerophytes that perform CAM pathway, and they are stem succulents and leaf succulents, and the examples for stem succulents Opuntia and Euphorbia, and leaf succulents aloe and bryophyllum, and as the plant parts, and they are the aerial parts, and they are succulent, and they hold water in the form of mucilage, okay, to face the xeric conditions. But if they open the stomata during daytime, what happens? And that water may get evaporated. So, in order to store and secure that stored water, and they open the stomata during night time. So, such kind of stomata found in the plants like Opuntia, Euphorbia, aloe, bryophyllum, and they are called scotoactive stomata. What is the problem with scotoactive stomata then? Why they developed CAM pathway? Let us follow the logic. And here the plants, they show scotoactive stomata, night open stomata. So, during night time they should uptake carbon dioxide, but at that time they can uptake carbon dioxide, but to perform the Calvin cycle reactions, assimilatory power, particularly reducing power, that is required now. So, the CO2 which is taken by the plants during night time is not immediately reduced to carbohydrate due to lack of assimilatory power supply in the absence of sunlight. So, they has to wait, okay, and so, and they these succulent xerophytes, so that they find another way, they fixes carbon dioxide during night time and convert that carbon dioxide into oxaloacetic acid and then to malic acid, and fix carbon dioxide in the form of acids, 4 carbon acids, and the process is called acidification. And later during daytime that acid is deacidified, undergo decarboxylation and release carbon dioxide internally, and in presence of sunlight, as it is daytime now. So, in presence of sunlight assimilatory power supply is there. So, now that released carbon dioxide in the mesophyll cells undergo Calvin cycle reactions and involved in the synthesis of hexoses. Okay. So, and in order to follow that scotoactive nature of stomata, and they adapted ecologically with scotoactive stomata, and to support that scotoactive stomatal activity, and they developed this biochemical pathway as a better adaptation, and that is CAM pathway.